Hello and welcome to the fourth of our groups today. We will have another series of presentations and hopefully another discussion panel amongst the speakers. First off, we will connect with Professor Franco M. Buonaguro, the director of the Department of Microbiology and of Molecular Biology and Viral Oncology at the National Cancer Institute and the AIDS Center. Professor Buonaguro, are you with us?
Yes.
Oh yes, we can hear you, and thank you very much for taking your time to join us. And please, without further ado, the stage is yours.
Okay, so my presentation will be on SARS-CoV-2 evolution and pathogenicity. And I will start just describing a little bit the situation since March 3, when actually Italy recognized that was the first European country in terms of COVID-19 disease. And as you can see, in few days, this is 3 March and 4th of March. So we moved from 2,500 to over 3,000, with the epicenter in the northern part of Italy. So specifically it was in the Lombardia region. And this is the situation on March 8th, when actually we had the national lockdown, so that all people were not able to move any longer from one region to the other. And this is just the initial part of the pandemic in Italy, because in a few months later, so by May actually, we had almost the end of the first wave. So from the molecular point of view, we can see something very peculiar. So this is on the 3rd of March, and what we see essentially is that the 2 isolates from Italy were from both of them were the Chinese clade, both in the Spallanzani is in Rome, where actually the 2 patients were Chinese traveling to Italy from Wuhan, and then we had the other one isolated in Milan. But just a few days later, we had the first Italian from Codogno, who was related to the German clade. So this is actually very interesting that we had the 2 imported cases, one from China, directly from China, and one from Germany. And so this is just to see what happens a few months later. This is April 5th, and we see that Spain is increasing the number. Italy is the second one. And then actually the whole pandemic moved to United States and Southern America, as it is now further expanding, besides obviously India and Brazil. In Europe, instead, we are observing the second wave in many countries. This is the after or the end of August, and we can see that there are the second waves in many other regions. What is very interesting from the molecular point of view is that essentially what we have seen, so this is now the graph from the same platform, where actually instead of just 11 types, we can see at the moment hundreds of them, and they are divided basically in 5 different major clades. And this is just the traveling roots of the different genotypes. Now what we see is that essentially most of the types are not anymore the Chinese. So 19A, 19B are the original Chinese from one region, and instead the 20A, B, C are actually the one in the Northern Europe. And these now are the beginning, the new emerging clade, which are gradually expanding, the one in pink. Going back to the evolutionary origin of SARS-CoV-2, what is very interesting is that actually there is something very peculiar about the spike region. The homology is not very close to the pangolin, which in some stage was considered to be the intermediate between bats and humans, but the highest homology which we have on the first line is of the bat Yunnan 2013, which was isolated from workers in a specific cave in China. So this issue is actually very unique because although the Western countries are not discussing any further this issue, it is a major issue within the Chinese community. And in fact, immediately after the paper on Nature on February 2020, we have some further commentary and data showing that most of the homology is between rat G13 and the one human beta CoV virus without any major intermediate. And this has been observed also studying with the homology search between the human, the bat, and the pangolin. And again, the closest homology is between the human and the bat. And there are articles always within the Chinese community where there is a major concern on the identification of this bat coronavirus strain rat TG13, which actually seems that has not been successfully isolated from the humans. So suggesting that some artifacts was actually going on in that region. Moving to different aspects, this is the evolution of the different genotypes, and in particular the variant D614G within the spike. And what has been very clear since the discovery is that essentially the G614 variants is very highly infective with the production of a very high quantity of viral particles, and the histogram shows that essentially from the middle of March, most of the types were from the G614, the one that we know at the moment that is much more effective. And this shows very clearly what was the story. So at the beginning of March, in Italy, we have the two types. So one is original from the Chinese, the two Chinese at the Spallanzani Hospital from one region, and we can start to see one G614, which was already prevalent in the area from Switzerland and Germany. And then a few days later, in March actually, and from the Codogno epidemic, what we see is just the the G variant. And we can see in this slide that the D variant, the original Chinese variant, was for very little, for just the few Chinese. Instead, mainly in Italy, we had the epidemic of the G614, which is very similar to the situation in France. And in Germany, we see the switch from the Chinese to the German type. And this is on the world scale, so where we are reported the African, the European, Oceania, South America, and so on. And in all of them, we see actually the switch from the D to the prevalence of the G614. Besides the high infectivity, there is a lack of supportive data on the pathogenicity. Actually, this preprint article published on September 1, 2020, we can see that there is not any real major difference switching from D to G. And in this very accurate analysis, besides that, we can see also the utilization of oxygen and with or without respiratory support. And we can see that the bars are relatively similar. And the authors conclude that that they do not see a higher mortality or clinical severity, but the variant G614 is associated with the higher viral load and younger age of patients. So for sure, at the moment, there are several groups that are studying further genomic determinants of pathogenicity. So looking at what are the prerequisite genetic modification that will increase the transmissibility and the morbidity of the virus. And we just, there are several comparative studies where the comparison is between the current SARS-CoV-2-19, the SARS-CoV urbani 2003, the MERS, and the other circulating recombinants. But what is a very interesting aspect that I would like now to focus is the difference in the type of diffusion within the country. In particular, the situation in Italy, which was the introduction in Europe. Italy was not prepared, and the peculiarity of this G614 variant was very unique. Was the first in Europe, and as you can see, the epidemic was in a very short period of time. So we are talking about March, April, and this very acute phase, so that we do not see in other parts of the world, except probably for Spain, but the distribution was much higher. So what we have is that the percentage of death associated with the infection was very high, was above 10% in Western country, in particular UK, Italy, France, and so on. In Italy was the problem of the epidemic arriving at the very specific high rate in one region. And this also, if already the flights from Wuhan and the China in general were already totally halted since.
Excuse me, Professor, but I'm afraid we're nearing the end of time. So if you could please come closer to the conclusions of the.
Yeah. So essentially, that is almost at the end. So again, this is just to see something else very peculiar. So that in India, for instance, we have a lot of cases, but the percentage of death is very low, and we really do not know why there is this type of data. I would like just to mention at the end that as editor-in-chief of Infectious Agents and Cancer, we have opened a specific series on the impact of COVID-19 and particularly on cancer. And finally, that on October 12 or 15, so in less than a couple of weeks, we will hold the free virtual course on SARS-CoV-2 COVID-19 related disease. Thank you.
Thank you very much, Professor, and here's hoping we'll be able to meet again on this other online conference
Okay.
as the later discussion panel. And now we're about to meet our next participant. Let's welcome Dr. Leah van der Hoek,
Yes.
virologist from Netherlands. Thank you for your participation, Dr. van der Hoek, and please go ahead with your presentation.
Thank you. Yes, I will talk about seasonal coronavirus reinfections. And I studied this to know how often we can expect this for SARS-CoV-2, because that's the only way we can foresee the future. And there are some anecdotal evidence of reinfections by SARS-CoV-2, only one peer-reviewed paper, and it was a perfectly healthy person. So it might be common, or it might still be rare. And to give an educated prediction, we studied the seasonal coronaviruses. There are in total 7 coronaviruses in humans, 3 recent introductions, but 4 seasonal coronaviruses, and they have been there for centuries. And that's 229E, OC43, NL63, and HKU1, and I've depicted them here in the phylogenetic tree. And here you can see that they are phylogenetically quite far apart, but also biologically. They use different receptors to enter the cell. NL63 uses ACE2, the others use other receptors. So they're not biologically alike, and they're also not genetically alike, which means that they're all very different, and you can use them as model system. If there's something that they all do, we might expect that also for SARS-CoV-2. We wanted to investigate these seasonal coronavirus and reinfections for decades, so up to 30 years. But that's not possible when you take respiratory material and do it by PCR, because there's no cohort that has collected these kind of materials for years of the same persons. But there are longitudinal blood samples collected in cohort studies, and in that you can determine rises in antibodies as marker for reinfections. And we designed 4 ELISAs for each of the 4 seasonal coronaviruses, and we used the C-terminal region of the nucleocapsid protein as antigen to detect antibodies. First, we validated these tests, and this is in a validation cohort of the GRACE cohort, with lower respiratory tract infections with known cause, PCR confirmed, and then we checked if the antibodies are going up with infection, and this is 5 weeks after infection, and you see here the colored dots for NL63, for 229E, for OC43, and for HKU1, they're going up when you have an infection. Then we checked whether the signals were specific, and here you also see the signals are specific because all values except 1 are below 1, which means that the highest signal in rise in antibodies is to the virus that's infected. Then we went to the cohort study. These are healthy subjects followed for decades, and that's the HIV negative arm of the Amsterdam cohort studies on HIV-1 infection and AIDS. And they're HIV positive and HIV negative persons followed, and we took the HIV negative persons, so they're healthy. That started already in the mid-1980s, and every 3 to 6 months a blood sample was collected. And that is up till now. So for some, we have 35 years of follow-up. Then I have examples of how it looks like when you measure the antibodies to coronaviruses. So this is one example. This is one person. On the x-axis, you have the years of follow-up. And on the y-axis, in the panels, you have the different viruses. So in red, there's NL63. And you see here a peak with an infection, 3.1 rise in antibodies. Here's another peak, So in total, 4 infections, and that's in 1987, 1996, 2007, and in 2013. The same for 2 to 9E. There are 3 infections, 1 OC43 infection and 3 HKU1 infections. And of note, these rises and lowering in antibodies, this is not something that can simply occur when you have no infections, because in gray here in the bottom panel, you see the antibodies to measles, and you see that that's a perfectly flat line in gray. So if you have no infection, your antibodies levels are stable. And the same you see here for another example. Also here, this subject number 9, completely flat levels for measles. And here the peaks 6 times for NL63, 3 times for 2 to 9E, 6 times for OC43, and here an HKU1 infections. And in total, we did this for 10 persons. And then we checked whether these peaks in antibodies are associated with symptoms, because these 10 subjects, they reported with every blood collection whether they have had in the previous periods an influenza-like illness. So they self-reported whether they had fever or cough or sore throat. And here you see that with any virus, that's any coronavirus, and any symptom, any influenza-like symptoms, that we have a significant association, which means that these reinfections really occur with symptoms. Then we go to the most important slide of this presentation, and that's the time between infection. And here on the x-axis is the time between the infections. In total, 50 infections. We could determine the interval between one infection and then the subsequent one. And we saw already that for some, we had already after 3 months, after 6 months, we had the reinfections, and there were even more after 9 to 12 months. So this can occur very fast in 6 months or 12 months, and it happens for reinfections occur for all the seasonal coronaviruses. So here I end with my conclusions. The capacity to reinfect is a general characteristic of the human coronaviruses. This reinfection can occur frequently 1 year post-infections and sometimes shorter. And if we translate this, SARS-CoV-2 has settled itself in the human population, so we probably can expect that it will behave the same and that we will get reinfections with each subsequent wave, and this might be with symptoms. The acknowledgement, this study was financed by the EU, an innovative training network called HONORS, which I coordinate. I thank the Amsterdam cohort study applicants, of course, because of every 6 months or 3 months donating blood, reporting their symptoms. And this is a great source of specimens and a huge and very important collaboration. And Arthur Edridge really coordinated the study, and he's the first author on the paper. And I thank you for your attention.
Thank you very much, Professor. And there's one question I'd like to ask you. So, on one hand, as you've just shown, reinfection is quite frequent. However, on the other hand, from what I believe we know so far, COVID-19 mutates relatively slowly and to a relatively low degree for a virus of its type. So once we arrive at some pharmacological solution, be it a vaccine or some other drug, do you think we can realistically expect a solution that will stay relevant, or should we think of it more of like the common cold, where every season there's a new strain and we basically start all over again?
Well, I need to correct you on that. These reinfections, they occur with the same virus. So these human coronaviruses don't mutate fast. So they're not like influenza. So they mutate as fast as SARS-CoV-2 is doing. So these infections are infections with the same strain, and not because they escape immunity like influenza.
Oh, thank you for that clarification. Then, if I may follow up on what you've said, while we know coronavirus is quite different to, let's say, the previous SARS epidemics of past decades, do you think there are some similarities to the contagious viruses we already know of that could be useful in steering our approach to it? Well, we still have to discover the exact solution. Is there something we can base on the viruses we already know how they work, how we dealt with them?
Well, it's very important to determine how ill you get, because that's the thing. I am quite convinced that reinfections are going to occur, but a first infection might be completely might be different from repeated infections. And what we saw when we saw signs and symptoms, these are normal influenza-like symptoms, like common cold. So a first infection might be completely different and more severe than a subsequent infection. But that's really something that we need to find out. And let's assume that second and third and fourth infections are more milder, and that this virus will start to behave like these common cold viruses. Remember that these 4 seasonal viruses, they also came from animals. So they have been introduced 100 years ago or hundreds of years ago, and they have settled themselves, especially exactly the same as SARS-CoV-2 is going to do.
Okay, so one more, I suppose, given this data about the possible reinfection rate, I suppose the idea some scientists have posed about basing a possible medicine on the blood plasma of people who have recovered from the virus, you'd probably be somewhat skeptical of it, given the frequency of reinfection?
No, indeed, there is frequency of reinfection, but that coincides with the lowering of antibodies. So we see these like I showed these peaks. You see that these antibodies drop very rapidly. So here, these antibodies are probably protective. So the passive immunization that people are planning with plasma from people who have experienced COVID-19 will definitely be an option, because then they are still protective. But it's just the problem with reinfection is that you lose these antibodies so quickly.
Thank you very much for answer, Professor, and for participation in general in our conference.
Thank you very much.
Thank you very much. And now we'll be off to meet our next guest. Yes, we are currently trying to connect with our next guest, whom I believe to be Professor Kari Stefansson from the Iceland's National Academy of Sciences, and also founder and CEO of the genetic engineering. I might have said too much, of a biopharmaceutical company, Decode Genetics. Though I believe the first person we'll actually reach would be Professor Rushika Perera from the USA, representing the Department of Microbiology, Immunology and Pathology of the Colorado State University, and also member of the American Society for Virology. I already saw Professor Perera for a second, so I believe we should be connecting with her in about a minute. Welcome, Professor Perera. Professor Perera, can you hear us? And also founder and CEO of the genetic engineering. Welcome, Professor Perera. Of a biopharmaceutical company, Decode Genetics. Yes. Sorry, can you hear us? Because I'm afraid the connection can be a little choppy from what we were seeing. So I believe the first person we'll actually reach would be Professor Rushika Perera from the USA, representing the Department of Microbiology, Immunology, and Pathology of the Colorado State University, and also member of the American Society for Virology.
Yes, can you hear me?
Yes, we can hear you now. We've had a slight error in the meantime, but I believe we're already connected. We can hear you well, and we're very, very glad to have you here with us.
Great. Thank you for having me, and I'm glad to be part of this conversation.
I'm sorry. If you have the stream in the background of our conversation, could you please mute it because it echoes on the...
Oh, okay.
Can you hear us?
Let me try.
And also founder and CEO of the genetic engineering. Welcome, Professor Perera.
Okay, is that better?
Yes, I believe that has solved the issue. So thank you again for participating, and without further ado, please present your speech.
Okay, thank you again. And what I'm trying to share with you today is the work that we're doing in the antiviral space. We've been screening at Colorado State University. We have a large biosafety level 3 facility that we have set up to screen antivirals, and we've been working with universities and companies around the world to screen their inventories. And some of these antivirals have been investigational compounds and FDA-approved drugs, as well as disinfectants, inhalants, and biologics. So I'll just present some ideas of how we have gone about doing this antiviral screening. So we have 3 phases. One is to establish basic assays to screen a large inventory of these compounds. Phase 2 has been to really develop some synergy models for combination therapeutics. And then phase 3 is some of our innovative ideas of how we should really go after looking for antivirals and combinations of antivirals that would be more effective. So for the basic assays, we've been using SARS-CoV-2 isolated from the USA, and we've been using cells both from African green monkey as well as human cells and primary cells. Our antiviral assays have included high content imaging for cytoprotection and cytotoxicity, as well as plaque assays for viral entry and infectious viral titer. We can also measure genome amplification using qRT-PCR. Once we screen in vitro, we have the capability to drive into animal models and test in the hamster as well as deer mouse and bats with our colleagues at Colorado State. So I'll give you some some basic data from the basic antiviral assays that we've been testing. This is on disinfectants and inhalants. So for the disinfectants, we've been using plaque reduction assays. So we mix the disinfectant with the virus, and then we look for how much infectious virus is left. And this is a disinfectant on the left that we've tested against SARS-CoV-2, and it has now gone on to FDA approval. On the right-hand side is an inhalant where we test the inhalant against the virus by mixing it just like a drug, and we look for reduction of virus titer. This inhalant is important for lung diseases and now has gone on to phase 2 clinical trials. We also have developed the capability for visualization through high throughput imaging. And I'll show you an example of a biologic. So in high throughput, high content imaging, we can image the entire 96-well plate, and we can look at live and dead cells as well as total cell count. And you can also look at cell morphology, position properties, as well as confluence. So we use this to triage several antivirals. This is an example of a biologic, so natural product. And what you see here on the left is in green are live cells, in blue are dead cells, and in brown is viral titer determined by plaque assays. On the x-axis is increasing concentration of drug, and on the y-axis is cell count. And so I have highlighted a concentration where you see cytoprotection by the compound, where you have higher amount of live cells, less amount of dead cells, and less virus. And on the right-hand side is the corresponding plaque assay showing that that same concentration reduces virus titer. So we're using these assays to screen lots of compounds, but I also want to really emphasize the importance of combination therapeutics. As you know, for HIV and HCV, it's been very, antivirals have been very productive in terms of controlling the disease. And so we've been developing with our industry partners combinations of drugs that can be used against SARS-CoV-2. And I'll show you an example. The reason combinations are really important is that if you're using a monotherapy, so a single drug, you have to have a high dose, right? As we know from remdesivir and hydroxychloroquine and other examples, you have to have high doses, and then you also have complications. So complications include idiosyncratic complications, which means that each individual will respond to drugs differently. You can have hyperreactive, or tachyphylaxis, which means that the individuals will stop responding very quickly, suddenly. You have impact on comorbidities. And so combination therapies are a way to go about really strongly attacking some of these complications. So this is an example of a combination therapy that is now in clinical trials. Drug A is directly targeting the virus, and drug B and C are targeting the host. And so if you combine all three drugs, you can have multiple effects. One is additive, that all three drugs will add to each other's efficacy. You can have antagonistic interactions, which means that drug A might be interfered with by drug B and C. And then you can have synergistic, which means that drug B and C enhance the efficacy of A. And what we see here is in the graph, you can see that in blue is virus control, in orange is drug A, in yellow is drug A plus B, and then in green is drug A plus B plus C. And we have a 2.5-day delay in viral infection with the triple drug. And on the right-hand side, you can see that we get a 6-log decrease with 3 drugs versus a 3-log decrease with 2 drugs, and 1 to 3 logs between single drugs. What we also discovered from this triple combination is that drug B and C definitely reduced the EC50 of drug A. So the amount of drug you need to accomplish the goal is much less. So it's a very powerful way of going about it. And so finally, I just want to throw out some ideas that we are pursuing to really identify how we come up with combinations of therapeutics. So my laboratory is really focused on metabolism. And one thing we need to understand is that the human organism is impacted physiologically by disease vectors, microbiome, pathogens, vaccinations, and the environment, and also therapeutics. So your physiology changes. In the past, we have been studying these physiological changes using genomics and transcriptomics and proteomics. So looking at how the genetic structure of the organism changes. But what we really need to be looking at is metabolites, because metabolites are the intermediates and products of biochemical reactions. And so you really need to be looking at how the physiology changes and produces alterations in metabolites. So what is important also to understand is that most drugs and inhibitors are metabolites. So if you study the metabolome of an organism, you can actually identify natural inhibitors and drugs that can then influence your design of combination therapeutics. So using these ideas, we're using systems biology-based therapeutics. We're trying to understand how drugs interact with each other, how drugs metabolize in the human body, and what those metabolites, how it impacts the host, how co-infections and the microbiome may alter how you might respond to a drug. And in the end, what we have to understand is that not every drug is good for every individual. So our goal is to come up with companion diagnostics so that we personalize medicine for each individual. So this work is being done by several people at Colorado State. My team includes Elena Liang, Gabriela Ramirez, and Carly McAllister, and then also my colleagues Brian and David. And the funding has been from the Becher Foundation and also Colorado State University. Thank you.
Thank you very much, Doctor. Could I ask, is there any possible time frame you could estimate in which we could expect these ideas turned into actual pharmaceutical product available to public?
Okay.
Oh, sorry, we've already with our next guest, Professor Larry J. Anderson from the USA. I hope I'm not wrong based on your expression.
No, I was looking at a different screen.
Oh, great. I was afraid for a second. So yeah,
No,
let me
no.
quickly introduce Professor Anderson, an American virologist who has held senior positions at the CDC's, that is Centers for Disease Control, for over 3 decades. For example, directing the program following the SARS outbreak and serving as the special advisor on smallpox. Thank you very much for joining us today. We are very honored, and I'll shut up and without further ado, let you have the stage.
Okay. Well, what I'm going to do is not talk about the virology so much, is talk about the complexity of the impact of the pandemic on health and community.
I believe we might have a slight connection issue going on right now that we are working to resolve as we speak. We are not yet quite aware of the cause of the problem. It might be a problem with connection itself, or perhaps even simply a device crashing. So, oh no, we got a confirmation that it's a case of internet connection. So, please let's just wait a minute so our professor can probably reset the router or something like that. Glad to have you back, Professor.
I disappeared for a while. I'm not sure if that was my end or your end, the instability, but okay. So back to what I was saying, I guess. The complexity. There's the social economic side of things, and then there's obviously the health side of things as well. And at least in our experience here, they've often been viewed as competing interests, which I think sometimes that's true, but I think as we learn more, that's not actually the case. But in the social economic side, you've got obviously the impact on businesses, interactions, schools, colleges, everything in society has been turned upside down. And with that, a concern to how to rectify that, and also the health and long-term social economic impact of control measures. I think we're probably just beginning to appreciate that, and over time we'll learn more about that, and I think it's going to be more than what we expected. So that's one side of it. Then the other side is the health. There's the disease, the lost time from work, hospitalization, mortality, morbidity. And now we're learning that there's also long-term effects, possibly, certainly persistence of symptoms and likely long-term complications as well, although that's something that we'll learn more about with time. So the impact is pretty phenomenal. The other thing that I think we've learned in the course of this outbreak is that control measures work. Home isolation, shutting businesses, closing down restaurants and schools really does stop or limit transmission. Doesn't stop, but limit transmission very effectively. The other thing we've learned is that, and you know, there's the government and the public health side, with the public health side that I'm thinking about being the case identification, contact identification and management. It is also effective in containing or controlling the outbreak. But I think one of the things, and that's something we've kind of known. Now, how effective depends on the transmission, how we can identify cases and so forth. And with COVID, that's been a challenge. The other thing that I think we've learned is that the personal preventive measures, wearing masks, watching distance, keeping social distancing, washing hands, and staying home from work if you're sick or suspect that you're infected also is effective. And I think the piece that's probably become more appreciated in the course of this outbreak is the mask wearing part. The other thing that's very clear is, how do you get effective compliance? The messaging part, and getting people to listen to, understand, trust, and then implement what you want them to do. And that has been, in a substantial portion of the population here, a real challenge. And there's different reasons for that. Certainly some of the leadership challenges have contributed to that difficulty, and a major contributor to that difficulty. But in that context, and probably even in other situations as well, raise the question, should we think more creatively about who can tell messages, or who can help, and how to convince people to participate in the messaging to help get the compliance needed to have effective control or containment of an outbreak. Now, getting messaging and compliance, or trust and understanding and compliance with messages, is true in many settings outside of a pandemic. Certainly, vaccine hesitancy creates a problem, is one setting.
I'm afraid the connection errors have returned again, but as we've last seen, this had not taken long to restore. Here's hoping we can again resolve it immediately, and if not, and let's wait a minute and see if that's the case. But even if we lose Professor Anderson, which yes, I'm afraid is what will happen, we really hope to resolve this issue in time for the possible discussion panel. And now let's welcome Professor Tamir Abdelrahman from Luxembourg. Can you hear us, Professor?
Hello. Yes. Hi. Yes, I can hear you. Yeah, I just yeah. So roughly, I'm just going to talk today about the efforts that we have been doing actually in developing this kind of genomic mapping. I don't know. I'm trying to share my screen.
I believe we are seeing it. I'm seeing the presentation you've prepared, so it should be working.
Okay, so basically it's just about that we developed a kind of a protocol for whole genome sequencing using amplicon sequencing approach with Illumina based on the ARTIC protocol. And that was really very important for us to have a kind of an idea about the circulating variants in Luxembourg. We had the difficulty to have a whole genome sequence of low viral load samples. Just to give you a glimpse of the data that we have, so we went through all of the clinical mutations that are reported in the literature that has a clinical significance. The most widely reported one is the D614G, which is mainly related with higher infectivity and higher case fatality. And the other one that we have seen here in the circulating lineage in Europe in general is the B323L, which is linked with the higher severity as well. So what I wanted to share with you is just that what you have over here. So this is a representation of all of the circulating variants that we had sequenced so far in Luxembourg. We had done around 500 sequences so far, and we can see, I don't know if you can follow the pointer over here, that there are clusters, pan-European clusters, I would say. So we have seen a cluster with Austria, of course, a cluster with our neighbors, Germany and France, and another cluster coming with Scotland and UK. So that's very kind of interesting, finding that the clinically significant mutation, which is D614G, is very much prevalent in the Luxembourgish population. So 95% of the samples that we have sequenced had been carrying this mutation. Although the limitation over here, and I'm sure that you share that with me, is that we don't have access to all of the clinical data, so we can make kind of association studies and try to identify new mutations. But what is a common thing in Europe is to have the B1 lineage, which is the prevalent lineage in Europe. That was the same case and the same situation in Luxembourg, not much different from the rest of the Europe. Interestingly, we have a couple of variants from the A2 lineage, which were mainly returning traveler from Spain. And it does match the data that we have seen from Spain having the lineage A2. So that was a quick glimpse. I've been asked to talk very shortly and very briefly about the circulating variants over here in Luxembourg. I'm looking forward to share discussion about what is happening in other countries and what are your experiences. But in a way, over here in Luxembourg, I'm representing the national lab in Luxembourg, and we're really keen to collaborate in improving the knowledge about the sequencing and the variants from the microbial genomics point of view. So please feel free to contact me if you have any kind of you need any access to sequencing facility, and we would be looking forward to collaborate together to understand more about this virus. So I assume that there will be a discussion later on. So I'm just back to you, and thanks for your kind invitation.
Yes, thank you very much, Doctor. And yes, I do believe unless we happen to run out of time because of the some delays and technical problems, we will hope to have another discussion after all the presentations from these groups are over. So stay tuned, and yes, hopefully there will be takers for the genomic data.
Okay, brilliant. Thank you. Bye.
Thank you very much, Doctor. Now let's wait for our next speaker, Matthew D. Moore from the University of Massachusetts.
Oh, okay. Okay. So will I see anything?
Yes. Welcome, Doctor. Can you hear us?
Yeah. Sorry about that.
Welcome, and we're very glad to have you. And please, the stage is yours.
Okay, great. Yeah, thank you very much for the opportunity to talk. My apologies about the scheduling misunderstanding, but my name is Matt Moore. I'm a professor in the University of Massachusetts in the United States in the Department of Food Science. So I've actually run a lab that focuses on applied environmental viruses, and there are a number of aspects related to sort of those paradigms that we are actively in the process right now of trying to apply to SARS-CoV-2. And so sort of the first thing that's a specialty of our lab is trying to understand the sample processing portion of detection. So especially with respiratory pathogens like SARS-CoV-2, trying to gain an understanding of areas that are higher risk in terms of airborne presence of the virus. You normally do environmental sampling through an air collector. And so we've come up with, and we're doing research on ways to better understand how to process samples so that you can reduce the amount of inhibition to detection, and you can maximize the amount of virus in your sample when you capture it and concentrate it, whether or not you're dealing with an environmental sample or a clinical sample that may also contain inhibitors. So that's one of the areas that our lab is actively working on, and that we're open to collaborating with folks on. Another area that we're actively working on trying to apply a number of techniques related to SARS-CoV-2 is the development of an application of a number of novel portable detection assays that we actually have been developing for other viruses or other RNA viruses that we're in the process right now of applying to SARS-CoV-2, including one that's actually nanopore based that can provide not only portable and rapid detection, but can also provide subtyping information. So there's sort of an example, when you add your virus to a sample, you'll get a characteristic fingerprint, and that fingerprint is actually so specific that it can be specific to different subtypes. Similarly, we've been developing a portable isothermal amplification technique called recombinase polymerase amplification in terms of applying it to SARS-CoV-2 samples. And so here's sort of a picture of a really great example from a group that developed a similar assay for influenza virus, where you're able to fit everything from taking somebody's sputum to getting a sensitive result in less than 30 minutes. And you can fit everything in a suitcase. You don't need an electrical grid. Another area that our lab is working on, even though it looks like fomites are not really, at least to this point epidemiologically, are not really a major vehicle for SARS-CoV-2 transmission, we're trying to better at least gain an understanding of not just SARS-CoV-2, but how other enveloped and non-enveloped viruses are transferred between different surfaces. So it's one thing for a virus to survive on a surface, but then that actual transfer efficiency and its ability to transfer and survive that transfer process hasn't been very well characterized for a lot of viruses. And so this is another area that we're interested in, even though for SARS-CoV-2, like I said, it doesn't look like fomites are a major vehicle, but still understanding their potential for transfer is needed. And then another area that we've had a number of projects on is developing better inactivation agents, not just for SARS-CoV-2, but for other viruses as well that are not just respiratory, but also foodborne. And so if we can better create inactivation agents that are not only really efficacious, but that are also environmentally friendly and potentially not very cytotoxic to humans, that's what we're focusing on now, is better structuring things that we believe are safe or not very toxic to humans, but that can destroy viruses. Additionally, we're looking into ways of better designing materials that are green, that can actually capture and kill viruses as well. And so I just wanted to give like a really large or really broad overview of those are the types of projects that we're doing. And I just wanted to put that out there and wanted to thank you all for allowing me to talk. And I just wanted to sort of mention that our lab is really open to collaboration. And so if any of these topics seem interesting to you, or if there's a project that you have that you're looking for a partner on, I just wanted to put my contact information out there. Our lab is always open to collaboration, and our department as well is always open to collaboration, as we have a very international department. So please feel free to email me if you wanted to discuss anything that I just mentioned, or you have a potential project that may involve implied virology. And so thank you very much for your time.
We'd like to thank you very much for participating in this meeting. It's really a shame we couldn't do a physical conference for obvious reasons, because those far recent topics just beg to be shown and touched. But alas, not this time.
Yeah.
So yeah, thank you for taking your time. Hopefully during the discussion panel someone will take you up on that offer. And right now we'll have to move to our next speaker, Professor Jesus El Romalde from Spain and the University of Santiago de Compostela. Welcome, Professor Romalde. We are very glad to have you.
Good evening.
Yes, and please, at your leisure.
Okay, my name is Jesus Romalde. I'm from the University of Santiago in Spain, and I'm going to talk to you about wastewater-based epidemiology for COVID-19 in the next minute. Soon after the beginning of the COVID pandemic, it was demonstrated the presence of SARS-CoV-2 in the feces of the patients. And you can see in this table that even the shedding of the virus in the feces can last for several weeks after the disappearance of the symptoms, after the recovery. And at the same time, it was detected also that this shedding was occurring in asymptomatic individuals, and that the virus of the viral RNA can be detected in sewage. Of course, probably the virus detected in sewage is non-infectious viruses, as reported by the team from La Charité at Berlin. But anyway, the detection of this viral RNA in wastewater can be a useful tools to monitor the circulation of the virus in the communities, and also as early warning system for the disease. So this is a schematic for the wastewater-based epidemiology. We need to sampling the wastewater samples, then concentrate the virus from this wastewater, do the viral quantification in these samples, and then we have to know the shedding rate, the viral shedding rate from each individuals perform the normalization for population to determine the infected individuals per capita, and then do the epidemiological modeling to report to the public health to take decisions. The critical factors for the wastewater-based epidemiology implementation are shown in this slide: sampling, virus recovery, virus quantification, and we will see very fast any of those. Sampling, we have to consider the best time of samplings to be really representative of the population, and we have to consider also which is better, the composite samples taken during the whole day on grab sampling taken at that specific hour. Another important point is the viral recovery and concentration from the samples. There are different concentration procedures with diverse effectiveness, and we have to take into account that these concentration procedures have been usually developed for naked viruses and not for enveloped viruses, and SARS-CoV-2 can present some specific behavior. Regarding the virus quantification, there's a lot of primers and probe sets already published with different sensitivity and specificity for the virus. So this is a point that has to be taken in consideration when develop the wastewater-based epidemiology. As mentioned before, we had to calculate the shedding rate and relate this shedding rate with the levels of the RNA detected in wastewater. We can use different biomarkers for do the population normalization needed for the modeling. And of course, we have to take into account ethical considerations. Although it seems that has low relation with ethical risk, sometimes sampling a small population could lead to stigmatizing behaviors with this population. So we need to avoid such risks. Very fast, some results obtained in our study in Spain. In purple, you see the RNA, the viral RNA levels in the wastewater, and has to be related and even can be detected early before than the cases in the population. And very fast, this is a map of Spain and shows some of our results. You see the different dates from July 15 to September the 2nd, and how these are the confirmed cases in the different regions, and the stars indicate the detection in the wastewater. So you can see that with the different, according to the increase of number of cases in the population, we start to also to detect even as low as 3 or 19 cases in the province. We can detect the virus in the wastewater. So, and these are the levels of viral RNA of the different weeks. You see that from the start in July to the end of August, the increase in the viral RNA levels are dramatic. So to conclude, the presence of coronavirus in wastewater represents a new challenge and opportunity to employ the wastewater-based epidemiology. There is a need to optimize all the procedures, the recovery concentration methods for enveloped viruses. We know that we can use the presence of this viral RNA as an indirect population-level diagnostic tool. We have to be careful with the representativeness of the samples, the viral concentration methods, and the population normalization. And the final conclusion is that a well-validated wastewater-based epidemiology system is imperative for viral surveillance, particularly with respect to an early warning system for the present and next human pandemics. And thank you very much for your attention.
Thank you very much for your presentation, Professor Romalde, especially since it, honestly speaking, appears to be a very unique approach to tracking the epidemic. So we're doubly glad to have it shared with other colleagues. So the idea can truly percolate and hopefully help save lives, because that's what it's all about in the end.
Okay, Thank you.
So, thank you very much. And as for our conference, I believe we will be trying to connect once again with Professor Larry J. Anderson, as we believe that whatever was plaguing our connection has already been resolved.
Okay, thank you.
Thank you. Oh, yes, we are establishing the connection. I'm already seeing the professor, just not on the main feed yet. So this truly probably must be a case of just a final confirmation that it's all.
Do they really want me to continue?
Oh yes, welcome back.
Sorry
Professor.
about that.
Uh,
Yeah, our
I believe the
internet crashed. I
speech is
think
worth
it's
this
because
battle.
well, this time of day there's a lot of schools using Zoom, and I'm at home, and I should have gone to another site that had more. I mean, it's been good most of the time, but it just crashed then anyway. So now I'm on the cell phone internet access. So, okay. So I was just about ready to finish up, actually. Is it relevant? I mean, it's been such a long wait. Is there?
Well, I'm interested, so please go ahead.
Okay, well, so then the question becomes, how do we kind of build the foundation in the future that allows better ability to get people to listen to understand and trust the message? And I think there's a lot of different answers to that. One that I thought about is personal responsibility, that it becomes part of medical education, scientific education, public health education, for the students as they move into the professional career to be tuned in to the opportunities to communicate and participate in educating in schools, community, whatever. So that's kind of one piece of that. And there's lots of other pieces that it's kind of building the foundation or educating the future medical, public health, scientific consumer. Are there things that we can do that better? And if you think about here in the United States, there's groups.
I didn't understand that.
I don't know what that is. It's my cell phone doing something. That there are groups that have been messaging the other side for a long time. And in some ways, effectively creating kind of an anti-scientific, anti-vaccine community. Can we do a better job of building the foundation, educating the public when the opportunity arises? And the opportunity arises is actually very important. There are times when people pay attention. Talking about COVID right now, people pay attention, they're interested. And, when Ebola was happening, people were interested in that. When the first SARS was out, people were interested. But there's lots of other situations too, not necessarily in the medical, but in the scientific world, that there things come up and taking advantage of that. So that was kind of where I was ultimately going.
Yeah, thank you very much, Professor. And there's one thing I'd like to ask you to follow up on what you've said, because specifically taking the US as an example, I think what's quite characteristic is just how politicized the response to the COVID has become. And what I mean is that, in my opinion, for the most part, in the other parts of the world, while we're talking, of course, about somewhat mainstream discourse, when you've got the opposition to measures taken, for example, the quarantine measures, you've also, of course, always have the conspiracy crowd or whatnot. But usually, when there's opposition, it's like, I'm a restaurant owner. I don't want my business closed. I just don't care about it that much to accept it for the greater good. While in the US, from what I've seen at least, there's this additional layer of, hey, wearing a mask is a Democrat thing. So that's why I should or I shouldn't do it, because it's a concrete political act, apart from what it actually means as a measure that changes one's life. So I'd like to ask, given that context, do you think that what chance there is to really take lessons from this pandemic, the response, what worked, what not, and what's the chance that it will simply get swept under the rug as part of the general turmoil of the last years?
Well, that's a really good, and it's a tough question, and in fact, I don't know the answer. I think it has to do with leadership, and in the context of COVID, leadership, our president and some of his compatriots have really worked against the efficacy of control. So then you run the situation, can you get there despite that? And I don't know what the answer is. I think there are probably some things that you can do a better job, get around some of it. For one thing, it's in everybody's personal best interest to do this. If you want your kids to go to school, a big roadblock is community transmission. If you want to watch sports, and the sports teams, players don't get COVID so they can play, a big part of that risk is community transmission. If you want your job back, if you want to be able to go to work and the restaurant's open, bars open, whatever, a big part of the risk of that is community transmission. So everybody has a vested interest. And I think ultimately going on, it's good to help protect your neighbor will work for some people. And as you suggest, for a lot it won't. But if you help people understand that it's in their best personal interest, no matter who they are, what they are, to do this, then there's an increased chance. So how do you get that message across? Now, in Georgia and some other states, and many states, sports is a big deal. This is just one example. But the major sports players, the coaches of the major teams, at least in U.S. football, some of those have enormous potential influence within their fans and even to some extent nationally. And is there a way to convince them that it's in their best interest just financially to control community transmission? They can play and get people to come to the games and they can make more money. Well, okay, is that what it takes to do it, then that's fine too. So I mean, that disconnect between what leadership is doing and what's needed to control the outbreak is one of the reasons that got me down this path, because it's very frustrating. And it's, we were listening to The Splendid and the Vile. It's a historical accounting of Winston Churchill's first year in office. And the contrast in leadership, true leadership, is just amazing. You know.
Yeah, I understand.
And you know, telling the truth. And so I don't know the answer. I hate not to try. But I'm not in a position to maybe influence people. And I've tried to get there through other channels, but haven't gotten there yet. So that is a good question for everybody to bite on, because I do not have the answer.
Still, thank you for your attempts at answering. And yes, I do think that acting on the community level is, at the very least, a good start. So thank you very much, Professor Anderson. And now we'll meet with two professors from Cuba, Ms. Maria Guadalupe Guzman and Sonia Resic from the Pedro Curie Institute of Tropical Medicine, and they'll talk to us about the issue of COVID-19 diagnostics. Welcome, professors. Can you hear us?
Hi, how are you?
Hi.
Yes,
Many thanks for taking your time to partake in this conference on the other side of the world. And please begin at your leisure.
Can we start?
Yes, whenever you're ready, please go ahead.
Okay, well, thank you very much. I am Dr. Resic from Cuba. I am the head of the virology department at the Tropical Medicine Institute Pedro Curie in Havana, Cuba. I am going to talk about diagnosis, COVID diagnosis here in Cuba. Cuba, a Caribbean country with more than 11 million inhabitants, started to prepare its national response in late January. The plan for prevention and control of the COVID-19 include, among other aspects, the training of the healthcare workers, the enforcement of the national program for the surveillance of acute respiratory infections, and of particular importance, the preparation and further extension of laboratory infrastructure and facilities for the molecular diagnostics of SARS-CoV-2 infections. Cuba plan has four major characteristics: early or at least timely application of mitigation and suppression measures, massive and reinforced acute respiratory infection surveillance, enabling early detection of suspected cases, comprehensive contact tracing with rapid isolation of confirmed cases and contacts, and finally, the development of a rational testing program. In the context of the Cuban response to the COVID-19 pandemic, it is important to note certain pre-existing conditions that benefit the implementation of the national strategy. These include universal health care, the highest per capita of medical doctors per million of inhabitants worldwide, a well-structured primary healthcare system, and a previous history of facing emergency situations, for example, during the annual hurricane seasons. Also, the existence of a state-controlled economy and public health policies, expedited the mobilization of emergency resources, and facilitated the rapid isolation of confirmed cases as well as their contacts. In February 2020, a team of virologists from the Respiratory National Reference Laboratory of the Institute of Tropical Medicine Pedro Curie in Havana received training from the Pan American Health Organization in the diagnosis and treatment of COVID-19. We were ready when it arrived. On March 10, the first suspicious cases in the country were identified. 4 Italian tourists with respiratory symptoms in a hostel in Trinidad who had arrived by plane to Havana the day before. They were immediately transferred to the IPK on March 11, coincidentally the same day that the WHO officially declared COVID-19 a pandemic. Cuba confirmed the first cases of its outbreak. From then on, new infections were detected among foreigners or Cuban arriving in the country from Europe, Canada, the United States, China, and Latin America, and from Cubans who became infected in the national territory or being in contact with foreigners, or Cubans coming from abroad, or secondary cases related to foreigners. Cuba developed an active case detection in a large population groups, identifying the most vulnerable, the suspected cases, and contacts of confirmed cases, using both real-time polymerase chain reaction tests and rapid tests in some cases. The suspected cases and contacts are placed in isolation centers, while the positive individuals are hospitalized. The active screening is done at the health primary attention level and helps us to identify a high number of cases. Every time a case is confirmed, contact tracing is initiated to isolate contacts too. Molecular diagnostics of SARS-CoV-2 infections represents another key player in controlling the COVID-19 pandemic. The director of the World Health Organization highlighted the importance of testing on March 16 by stating, "You cannot fight a fire blindfolded. We cannot stop this pandemic if we don't know who is infected." So far, RT-PCR is an expensive test and requires a complex infrastructure as well as highly qualified and skilled persons. As well known, several Latin American and Caribbean countries lack the national infrastructure to develop a mass testing program to detect most COVID-19 cases. Global data show that in general, countries that develop a mass or at least rational testing program in timely fashion significantly reduce the impact of the pandemic by efficiently controlling viral transmission. Unable to perform mass testing, Cuba used an alternative scheme of molecular testing enabled by gradually equipment up to 12 laboratories in different regions of the country. The Respiratory National Reference Laboratory at IPK is responsible for the quality control of diagnosis made in all these centers. The Cuban SARS-CoV-2 molecular diagnosis program is following the WHO recommendations of gradually increasing the laboratory capacity for the molecular diagnostic of the virus and, more important, to perform daily tests to ensure that no more than 10% of these tests are positive. To date, the country has performed 606,400 tests after more than 6 months facing the pandemic. More than 5,000 people infected with SARS-CoV-2 have been confirmed, of which to this day, 4,000 have been recovered. We regret the loss of 122 lives due to this cause, but we are comforted and encouraged that no child, no pregnant women, no doctor, and no health worker have died. Cuba was one of the countries that traced all contacts. The Cuban approach of massive manual contact tracing was coordinated by the far-reaching primary healthcare system and the collaboration of thousands of medical students and healthcare workers. In combination with molecular diagnostics, a comprehensive program of contact tracing remarkably reduced the impact of asymptomatic and pre-symptomatic patients in quelling virus transmission. Cuba applied a functional strategy to control its epidemic based on the timely application of several mitigation and suppression measures, massive surveillance of acute respiratory infection, and the recently so-called COVID-19 Tetris, test, trace, and isolate. In addition, the recommendation to wear face masks in public places contributes to reducing transmission. Finally, the Cuban approach reinforced the epidemiological value of applying a rational testing program for the control of COVID-19 pandemic. Thank you very much.
Thank you very much, professors, for sharing your experiences with dealing with this struggle. And while now we'll have to quickly go to our next speaker to stay on schedule, I'd just like to make sure to remind you that after some 15 to 20 minutes, probably, depending on our schedule, there will be a discussion panel, and we'd really love to have you participate with others. So thank you again. And now we'll be talking to Professor Berlin Londono from the Kansas University, where he leads a vector laboratory. To talk to us about vector-borne disease surveillance and challenge during COVID-19 in Colombia. Hello, Professor.
Hello, how are you?
Fine. We're very, very glad to have you with us. So please start whenever you're ready.
Thank you so much for inviting me. So today I'm just going to talk a little bit about the vector-borne surveillance efforts that we are doing in Colombia and the challenges that we are facing during this epidemic. So here, just to talk a little bit about what we do in my lab, we have three big areas, but the main area of research is trying to characterize the salivary proteins in the mosquito vectors of dengue and malaria in a way that we can use these antibodies to a way to track vector-human contact. So what we do with these antibodies is that we try to define disease endemicity and risk. Then with this, we can evaluate vector control interventions and monitor the changes in exposure to these vector bites in people living in these endemic areas with time. With this, we want to influence the choice of control strategy in these areas. So our main goal really is to be able to map the level of these antibodies in a way that we can focus on interventions, reducing costs for the vector control agencies, and we can protect our community. Now, in the areas where we are currently studying dengue and malaria, we have a huge challenge on being able to detect who is infected with dengue virus and who is infected with SARS-CoV-2 virus. Why is that? Is because in these areas, most of them is just a health center, not a hospital. And the way that we are diagnosing dengue is by clinic. So we have a person with fever getting to the health center, and we send blood test. Now, what we are looking in that blood test is actually the platelet count and the lymphocyte count that will tell us if that person is a probable case of dengue. The problem with this is that several cases of COVID-19 present the same hematological findings, like low platelet count and low lymphocyte count, and on top of the fever. Now, to make these things worse in the areas where we are studying, for the past 2 or 3 years, we have an increase of dengue cases presenting respiratory symptoms. And so it makes it now very hard for us to can differentiate if we don't have the tests in these hospitals to say who is infected with dengue and who is infected with SARS-CoV-2. This is slowing our dengue diagnosis and is delaying the attention of these dengue patients. Now, it's important that to remember that in Colombia, we not only are facing a pandemic with COVID-19, but also dengue epidemic. So for us, it's really important. We need help in better ways to diagnose these diseases. Here's just to show a very nice review that I found where they discuss all the immunological factors related with the why these two pathologies or why these two infections can sometimes be confused if you don't have the right tools to diagnose the disease. And they are showing here both dengue and SARS-CoV-19. The COVID-19 patients may present gastrointestinal symptoms. We will have thrombocytopenia and leukopenia. So for that, we are really kind of looking for a better diagnosis test into having these areas where many laboratories are. The way that we are diagnosing dengue right now is through ELISAs, but because of all what is happening, the results for the ELISA will come back to the patient 10 to 14 days afterwards. By that time, the person or has recovered or is already in intensive care due the infection. Now, those SARS-CoV-19, SARS-CoV-2 virus and dengue virus, they induce what we call the cytokine storm, which will lead to endothelial dysfunction and may at the end lead to the severity of hemorrhagic manifestations in dengue, or will lead to the compromise in the respiratory organs in SARS coronavirus. So for us, what we want to stress is that we need better ways to diagnose these two diseases in the areas where we are studying right now. So for that, just want to thank my collaborators, and here is my contact information, and thank you again for inviting me.
No, thank you for participating and sharing your expertise with us and your colleagues, and we really hope to also have you on the upcoming discussion panel, which will come after one last speaker, I believe, which will be us connecting back to Cuba to Mr. Sonia Resic to tell us about her part of the Cuban experience with diagnosing COVID-19. Welcome again.
Hello.
Hello. Yes, so welcome again, Professor Resic, and please begin.
Well, is Professor Guzman, Maria Guzman.
Oh, Guzman.
I am the head of the Center for Research Diagnosis. No problem, no problem. And reference activity here at the Tropical Medicine Institute in Havana. My talk is to speak about the research portfolio that we are developing here in our country. As probably you know, the Cuban health system is free, universal, accessible to everyone, intersectoral, and with a wide participation of the community. It is based on the primary health care of a patient, and a very important characteristic of our health system is the timely introduction of the science advances. Very early, Cuba prepared its country plan for facing the pandemic, being finished at the end of January. Science and innovation play a central role in this plan and are crucial for the decision making. To respond to country needs, we have developed more than 300 research and innovation projects, supervised by the COVID-19 National Committee of Research and Innovation. Projects are evaluated by the National Ethical Committee, and clinical trials are approved by the National Regulatory Agency. Some of the principal research lines we have developed are directed to support viral diagnosis, the implementation of epidemiological and clinical policies for surveillance and control, the clinical management and the treatment of patients, among other related topics. Since March, Cuba has strengthened its COVID-19 diagnostic capacity, extending the molecular diagnostic from 1 to 13 laboratories and from 100 to more than 7,000 samples daily. To achieve this in only a few months, we had to evaluate and validate reagents, consumables, and equipment. Different RT-PCR platforms and extraction machines have been evaluated in our conditions, as well as reagents for RT-PCR from different origins and commercial companies, such as kits for gene E and P detection and or RF, etc. Additionally, we had to evaluate different transport media. The evaluation of rapid tests is also important, as well as the immunosomatic system for antibody detection. This evaluation has allowed to recommend to the Ministry of Health the best system for epidemiological studies. However, because the challenge of the low availability and expensive prices of some of these items, as well as the difficulties to arrive in due time, the country is deeply involved in the manufacturing of our own reagents, consumables, and equipment as part of a national project to introduce and restrain the molecular diagnosis. The swabs and trampos media manufacturing previous a careful evaluation allowed us to introduce them into the national surveillance for sample collection. Today, Cuba has developed a 3 serological ultramicro ELISA system for IgM, IgG, and anti-total antibody detection to SARS-CoV-2. These 3 systems were evaluated showing adequate sensitivity and specificity figures. Now these systems are being introduced for epidemiological studies to detect possible transmission areas. Another research diagnostic line is directed to support clinical and epidemiological policies. Initially, already COVID-19 confirmed cases were tested by RT-PCR at day 14 of symptoms. If negative, hospital discharge was indicated. However, after a study of a sample kinetic study in confirmed symptomatic and asymptomatic individuals, demonstrated us that at day 10, more than 71% of positive individuals were PCR negative. This result allowed us to change the first discharge policy. Today, all cases are studied by RT-PCR at day 9. If they are clinically recovered and have a negative PCR, they are discharged from hospital. After that, of course, they are passed to a surveillance and isolation in their own home. And after 15 days, a new PCR is done and is negative, they are completely recovered. However, in some individuals that remain PCR positive for weeks, we did a structural study using electron microscopy of nasal sample collected from symptomatic and asymptomatic individuals with a persistent positive PCR and demonstrate virus-like particles in a very large concentration and in different replicative stages. This result supported the permanence of them at hospital in order to avoid potential virus transmission. We did not discharge them, they stayed. Another investigation is directed to genetically characterize the circulating viruses, including those detected at the very beginning, at the middle and late of the Cuban epidemic, as well as some detected from asymptomatic and mild and severe symptomatic cases. The gene S sequence showed that a majority, 94% of the studied viruses have the D614G mutation. Today, we are advancing the sequencing of the whole genome. Additionally, the Barro sequences from infected individuals of interested epidemiological networks are under study. Immunological biomarkers study for clinical prognosis is one of the research lines. 3 pro-inflammatory biomarkers associated to disease severity and fatality were identified in the first nasopharyngeal swab collected in the first 3 days of symptoms. This result opened a window of opportunities for the treatment and clinical prognosis of cases. At present, we are involved in the identification of human genes associated with COVID-19 susceptibility or severity. Additionally, in mild and severe cases, we are also studying epigenetic factors such as environmental factors, medication, foods, to see the influence of them in disease severity. The kinetic of the viral excretion in nasal and nasopharyngeal samples, urine, sebum, and saliva is ongoing. One main research is devoted to characterizing the clinical manifestation of the disease, treatment protocols, and clinical diagnosis. Although we have followed clinical international protocols, Cuba has developed its own clinical treatment protocols, combining antiviral medication with immune modulators. One characteristic of our protocol is the inclusion of novel medication developed by the Cuban biotech industry, such as the pulmonary surfactant sulfasen, the itolizumab monoclonal antibody. This monoclonal inhibits the effector phase of immunity, and the domestic recombinant interferon alpha-2b, which has a direct antiviral and immunostimulant activity. We recently were applying a nasal formulation in risk groups and vulnerable people, as well as a combination of alpha and gamma interferons in patients. CIGV258 product, which regulates innate immunity and induces the control of inflammation without immune suppression, is also under evaluation with very good results. In most cases where this product has been applied, C-reactive protein, ferritin, and cytokine levels have markedly moved toward normal, and patients are recovering from leukopenia and thrombocytopenia with substantial improvement in lung imaging as well as early PCR clarification. For people aged 60 years and older who experience immunosenescence, we are also using biomodeling T and a transfer factor, both natural cellular immunostimulants with good results. Cuban scientific institutions are developing COVID-19 vaccine candidates. Among those, there is one, Soberana in Spanish, that is on the clinical trial now. This candidate is based on the strategy of the Cuban BCG vaccine. This is the first vaccine for preventing serotype B meningococcal disease that has a powerful innate immunity stimulant activity. Considering the importance of research collaboration for facing COVID-19, we would like to express our willingness to sustain scientific and health exchanges to promote collaboration among our institutions. We also want to thank international organizations such as the World Health Organization, the Pan American Health Organization, Medicuba Europe, Global Fund, PNUD, and institutions such as Berkeley University and the Institute of Tropical Medicine of Belgium, among others. Thank you very much.
Thank you very much as well, Professor, for your presentation and participation. And now we'll go to our next participant, Ms. Tommye Austin, Chief Nurse Executive of the San Antonio Clinic in Texas. As anyone who's worked in hospital knows that nurses tend to be the unsung heroes that make the entire establishment work, and Ms. Austin might just become the most influential nurse of modern hospital practice, as she's a developer and creator of a unique mask to compete with the popular N95 respirator that there's not nearly enough of. Welcome, Ms. Austin.
Thank you.
So, please begin whenever you're ready.
Hi, my name is Tommye Austin. I'm the Chief Nurse Executive here in San Antonio, Texas at University Health System. And on March the 19th, the Centers for Disease Control stated that healthcare professionals, including nurses, physicians, respiratory therapists, could use handkerchiefs and bandanas to protect them against COVID-19 if we should run out of N95s. And so N95, as you stated earlier, is a respirator that we use for airborne isolation. When we heard this news, we were alarmed. And so being a sewer and a quilter, I thought about the filtration efficiency of cotton and other fabrics that we normally use to make handkerchiefs and bandanas. And we knew that that was not an acceptable practice. And so as a consequence of that, some of my colleagues and I went to different uniform shops to try to find masks to protect our staff. Then there were none to be found. It was at the height of the COVID-19 pandemic. And so one of the things we did, we went to the local fabric shop and could not find materials to make an efficient mask. Then we went to the paint store, and the paint store normally had N95s, but they were sold out. Everybody had the same idea, and they went and purchased all the N95 masks, so we couldn't add to our supply from that standpoint. And so having the skill of being a quilter, I decided to look at certain types of N95 masks. I looked at the halyard, I looked at the 3M N95, and I decided that based on some of the feedback that I had gotten from the staff, that we would create our own mask. And my late husband was an individual who should have been an engineer, but he wasn't, had told me early on that AC filters had an electrostatic property. And I learned that the hard way by purchasing some filters for the house for our air conditioning system that did not have electrostatic property, and he was not very happy with me. And so that's how I took the time to learn about electrostatic AC filters. And with that knowledge, I went to Lowe's, purchased some 3M AC filters that we used for the filter for the homemade mask that we made. Here is an example of the mask that we have made and created for our staff. We have about 6,000 in stock, so that if we get another surge of COVID-19, we have these masks on hand. So what I can share with you is that we not only made these masks for our staff. I've had individuals who have had cancer, who have had other autoimmune issues want the mask as well. And I have sent those masks to them to help them in their time of need. Some of the other things that we've done here at University Health System to stretch the amount of masks that we currently have in the N95 supply is that we set up a reprocessing system using our hydrogen peroxide steroid cleaner. And so what we did with that is that we gave staff 2 masks per week where you could use the mask every other day. And what we would do is you would use a mask, an N95 mask. After you finished using the mask, then we would put it in a paper bag so that we could send it to reprocessing, our sterile processing department. And then you would get the mask back after 24 hours so that we can make sure that the hydrogen peroxide was removed from the mask and you wouldn't have those side effects of using a mask that was filled with hydrogen peroxide. So that's why you got 2. So you would put send one to the sterile processing department, then you would use the additional mask, and then we would do that for at least 4 cycles. And that way we were able to make sure that our staff always had N95 equipment available. Now, what I can share with you is that in creating this mask, what we did was we took halyard material, and halyard material is what we normally use to wrap our sterile processing surgical equipment in the OR. And so what we did was we took that material, and it is also the interior of the mask as well. And then in between those 2 layers is the AC filter. Our original mask used an AC filter. And so we made probably about 4,000 of those masks using the 3M AC filter. We later learned about a material called Filti from a company out of, I think it's Oklahoma, and it's called Filti, F-I-L-T-I. The remainder of our masks are made with that material. And so what that material does, it has N95 property, and they have since made a mask using their material. The mask became so popular around the United States and the world that one of the largest uniform manufacturers decided to partner with me to create a commercial mask that we could reproduce and share with the rest of the world. And right now, I received the first prototype of that mask from the manufacturer, and I'll put it on. It goes on like this. And as you can see, you have full coverage. You don't have that leakage of air. You got to have a tight seal all the way around. And I performed the fit test in this mask, and it passed the fit test. The next step that we're going to do is to work again with the Southwest Research Institute in an effort to test the mask for breathability and for filtration efficiency. So what I can share with you is that it's very important to make sure that your staff feel secure. That is one of the reasons why we did this initiative in an effort to make sure that our staff felt secure with the amount of supplies that we had. When you watch the shows on television and you saw how the nurses in New York and other areas had shortages of supplies, it made the staff here very, very anxious here in San Antonio and throughout the world, I'm sure. And so what we wanted to do was to make sure that our staff focused on taking care of COVID-19 patients. I made rounds as a nurse leader, and I would see the fear of our staff. I would see the fear in the eyes of physicians, respiratory therapists. And because there was not a lot of information known at the time in regards to this disease. And so the last thing we wanted them to do was to worry about what was on their face. We wanted them to worry about taking care of these patients. And so the initiative to create a mask as a backup supply was very important. But some of the other things that we did is that we had a call twice a day in our organization to make sure that we knew how much supply we had on hand at all times. So what we did, we created a personal protection equipment dashboard. And with that dashboard, we would know how much supply we had on hand at all times. And the goal of the organization was to have at least 30 days of supply on hand at all times so that we could reassure our staff that we would be taking care of them. We also set up an initiative to make sure that we used our PPE wisely. One of the things I shared with you was the reprocessing, but we also had a PPE toolkit so that for our screening areas, for when people came in to visit our facility, we knew that we would use the level 1 mask for them. We knew for some of our surgical areas and for our acute care units that did not have isolation patients, we used the level 3 surgical mask. And then again, for our airborne, when we knew that there was aerosolation going to occur, we used the N95 mask. And so what I'd like to share is that one of the things we did was we made rounds often in the hospital, not only during the day, but also at night, so that our staff felt real secure about the supplies and the visibility of the leadership team. Now, do you have any questions of me in regards to what's going on with the mask currently?
Oh, yes. First, I just have to ask the obvious. Do you have any inkling just how widespread the Tommye mask design is right now?
Well, we gave the design away online so that people could make it themselves. And so on one of the YouTube sites, I had over 300,000 hits. So the mask is very, very popular. I've sent the mask. I personally have sewn about 500 masks and given them away myself. We use the mask here in the organization. I know that there's a physician, particularly he wears his mask every day. I make them special for him because he has a larger head, so he gets a custom Tommye mask. But I can tell you that I've sent masks to New York, Connecticut, everywhere, almost every state in the United States I've sent masks to. I haven't sent the mask out internationally, even though I've had requests from Italy and other places. And that's because financially I've been doing it myself. And so to cut down on costs, I've stayed locally within the United States.
Of course. And well, since you're a mask specialist now, if we take one step back from strictly talking medical practice, because we all understand the need for PPEs there, is, in your opinion, the way we treat the mask regime right now satisfactory? And by what I mean, in most places around the world, yes, you generally have the requirement to wear masks in public places, but there are so many exceptions, like, of course, bars, restaurants, clubs, and so on. Frankly speaking, half of the offices probably take them off the second they step past the door. And do you think that what we're doing right now is, so to say, good enough? Or will we really have to step our game once the infection spikes again during the autumn?
Unfortunately, I do believe that people have become laxed in their practice with masks. And I've even been in environments where people will have a letter from a doctor stating that they can't wear masks because of this or that. And so what concerns me is that with COVID being such a communicable disease, we should treat it with the utmost respect. And so now we've loosened, like you said, we're allowing people to go to bars. Schools are now open. Less and less restrictions. And so I think that we should be even more aggressive with using masks. But then there's articles that come out that say, okay, there's a strain that no matter what you wear, you'll still get COVID-19, which I think is an attack against wearing masks. And there are also anti-maskers out there who hate the fact of wearing a mask, and they create havoc in restaurants, stores, and this and that to distract from the fact that you need to wear a mask in order to not only protect yourself, but to protect others. And so my thing is that my fear is that as we increase the amount of kids going to school, increase the amount of people that can go into restaurant and bars, that we're going to have another spike. And we're just at the point where we're getting control of the virus. And so that does alarm me that we're not as reverent as we should be to COVID-19, because it is still a disease that can and has taken over 200,000 lives of people here in the United States. And so we need to be reverent to that.
Thank you very much for your insight and your participation in our event here. And right now we'll speak to Dr. K.M. Jacob, the Chief Medical Officer of Marma Medical Center. And if you've been with us the whole time from the very morning here in Central Eastern Europe, you might remember him in one of the earlier groups. But for all the rest of us, we'll talk to him again right now. To maybe recap, Dr. Marma, apart from being a practitioner, specializes in studying symptoms of the diseases with a particular interest in temperature. And temperature as it relates to COVID-19 and whether it's a reliable, and if it's then how reliable indicator of the disease, we'll learn in just a minute. Though I am getting information from the studio that there might be internet connection issues at this particular hour, still here's hoping that we'll manage to connect with Professor Jacob. Professor, can you hear us?
Yes, yeah, I can hear.
Yes, welcome again then. And well, without further ado, please begin your presentation.
It's okay. Fever is not a symptom in COVID-19. None of the disease require fever as its symptom. There is a sharp difference between the symptoms of fever and the symptoms of rising temperatures. Symptoms of fever include body pain, fatigue to mind and body, reduced appetite, reduced motion and indigestion, and aversion towards the colder substance, internal and external discomfort, etc. Symptoms of rising temperature include likeness towards cold items, like to drink cold water, feel discomfort while using a blanket, feeling hot, etc. There are no similarity between the symptoms of fever and the symptoms of rising temperatures. Fever never shows symptoms of rising temperature like decreased body pain, like colder substances, hate to hotter substances, feel discomfort while using a blanket. We can prepare a table showing symptoms, signs, signals, and actions of disease, fever, and rising temperature. This is the table known as the Jacob's table showing symptoms, signs, signals, and actions of disease, fever, and rising temperature. How can we separate symptoms of disease and the symptoms of fever and the symptoms of rising temperature? A fever condition means symptoms, signs, signals, and actions of both disease and fever are included. If we detect disease from fever condition, we will get the fever. So we can prepare an equation to separate it from disease condition. Fever condition minus disease equal to fever. Now I am introducing a table equations of separating fever, hypothermia, symptoms, signals, signs and actions from fever condition and hyperthermia conditions. This is the table. Okay, I will. Now I am preparing a table. So I have not enough time to rearrange them. So I am going to, immediately I am going to the symptoms, signs, and signals of fever. Or just 1 second. Like that, we can separate signs, signals, and actions of both the fever and the disease. Symptoms in fever condition, both the symptoms of disease and the symptoms of fever are included. If we deduct symptoms of disease from total symptoms of fever condition, we will get symptoms of fever. So we can prepare an equation. Symptoms in fever condition minus symptoms of disease equal to symptoms of fever. Signals in fever condition, both the signals of disease and the signals of fever are included. So we can prepare an equation. Signals in fever condition minus signals of disease equal to signals of fever. Signs in fever condition, both the signs of disease and the signs of fever are included. So we can prepare an equation. Signs in fever condition minus signs of disease equal to signs of fever. Actions in fever condition, both actions of disease and actions of fever are included. So we can prepare an equation. Actions in fever condition minus actions of disease equal to actions of fever. A hyperthermia condition means symptoms, signs, and signals and actions of both the disease and hyperthermia are included. So we can prepare symptoms, signs, and signals and actions of both the hyperthermia and the disease. If we deduct disease from hyperthermia condition, we will get hyperthermia. So we can prepare equation hyperthermia minus disease equal to hyperthermia. This is the table. Symptoms in hyperthermia condition minus symptoms of disease equal to symptoms of hyperthermia. Signs in hyperthermia condition minus signals in hyperthermia condition minus signals of disease equal to signals of hyperthermia. Signs in hyperthermia condition minus signs of disease equal to signs of hyperthermia. Actions in hyperthermia condition minus actions of disease equal to actions of hyperthermia. In fever does not show any actions of temperature rise. A new fever definition, diagnosis, and treatment is required. If a person who believes in conservative fever definition, diagnosis, and treatment, he cannot recognize these equations. How can we prove the fever is not a symptom in COVID-19? The fever is not a symptom in COVID-19 when examined in various directions. We can separate symptoms of fever and the symptoms of disease and the symptoms of rising temperatures. Like that, we can separate signs, signals, and actions of fever and disease and rising temperature. If we ask any type of question related to fever by assuming that the fever is not a symptom in COVID-19, we will get a clear answer. If we avoid or evade from this, we will never get a proper answer to even a single question. If we do any type of treatment by assuming that the fever is not a symptom, the body will accept. At the same time, body will resist whatever treatment to decrease blood circulation. Besides that, we use symptom definition. No further evidence is required to prove fever is not a symptom. The benefit of naming the fever temperature as a signal. The purpose of the definition of signal is to analyze the signal scientifically, to differentiate it and also brief it and understand it easily. If we say there exists temperature as a signal, one can easily understand without much explanation. In such a situation, automatically comes apt word as well as apt definition. That is why it is told to exclude the temperature of fever from symptoms of the disease. Conclusion. The fever is not a symptom in COVID-19 when examined in various directions. Temperature between 38 degree and 41 degree cannot be a symptom of any of the disease. None of the disease or cause of disease require fever as its a symptom. A different cause of disease like virus, bacteria, fungi, venom, horosine, and horror dream never shows the same symptoms. Opposite disease never shows same symptoms. Fever has been scientifically proved. Fever has never been scientifically proved as a symptom of a disease. Fever has the proper types of adaptation. Fever treatment should be revised according to the warning signal of a disease. A new fever definition, diagnosis, and treatment is required. It is wonderful that a physician having years of experience in fever treatment cannot understand actual fever definition and fever treatment have no relation with symptoms definition and diagnosis. In the early days of my discovery, I thought that fever is a symptom of disease. I never got any answers for even a single question about the symptoms. That induced me to find out the real truth of fever. I can eliminate conservative fever definition, diagnosis, and treatment. If you discarded this discovery, it would be an injustice to mankind. I also prepared 8,000 affirmative cross-checking questions, which can explain all queries related to fever. The real signs of fever can be termed as the physical. Can you allot me some more time? Hello?
I believe you can go for a short while if you need.
Yes, okay. I will start from. All the studies related to fever as a symptom of a disease have been done without knowing the purpose or temperature of fever is. 80% of COVID-19 patients have no fever. Then how can fever include as a symptom in COVID-19? Why up to 80% of patients with the COVID-19 virus have no fever? Is it because there is need for the care of fever? Is it because the disease is free of symptoms? Two questions arises. Most of the diseases may not have fever. Sometimes it disappears. Then is fever a symptom of which disease? Is the temperature between 38 degree to 41 degree centigrade can be a symptom of which disease? What is the use of symptom definition? Any or all other definitions, symptom definition should be describe the symptom scientifically. If it cannot describe clearly, there is no use of a symptom definition. Symptom definition is the only parameter necessary for a symptom.
I'm sorry,
symptom
Professor,
definition.
but I'm afraid
Okay.
we've got already another speaker on the line. Thank you very much for this presentation, and we hope to see you on the discussion panel later on after all the presentations.
Okay, thank you.
Thank you very much. And now we'll talk with Garwin Kim Singh, the professor of immunology and microbiology at Al Faisal University in Saudi Arabia. Welcome, Professor Singh.
I can hear you.
We can hear you, and we are very glad to have you with us on this event. So,
thank you.
Please go ahead.
Okay. Thank you for inviting me to join or participate in this working group on the COVID-19 pandemic. My name is Garwin Kim Singh. I've been in medical research, particularly in the field of viral immunopathology, for over 30 years. I've worked in different labs in South Africa, the United States, Australia, and Edinburgh, all on viruses and immunology. In 2012, I left research to take up a full-time position teaching virology and immunology at the medical school at Al Faisal University here in Saudi Arabia. What concerns me, particularly with this pandemic, is the link or the role of the scientific community and scientific expertise and the way it's perceived amongst the general population. And this, I think, is due to changes that have occurred within non-scientists, the non-scientific layman population, and in the scientific community itself. This big change happened to me in my impression in the mid-90s, when research funding started to get a lot tighter by governments due to budget constraints or reallocation of resources. And this budget cut also happened to educational institutions. Universities had to find new income streams And so they accepted more enrollments, particularly. This resulted in a huge number of PhD graduates looking for the same pot of research money and scientific jobs. A lot of them got picked up by industry, but with industry came more tightening of intellectual property and also more restricted collegiate cooperation between scientists. Coupled with that, particularly by the late 90s, was with the expansion of the internet. This meant that groups within the public who hadn't gone through scientific training could access information which previously was only of interest to those working in various scientific disciplines. This allowed for the rise of special interest groups with non-scientific agendas like anti-vaxxers, big pharma, and so on. And this has also done a lot of had a big negative impact on the respect for those who work in science, as well as unrealistic expectations that can come from the profession of science, of scientific research. And on top of that, by the beginning of the 2000, we then started getting a huge explosion of scientific journals, mainly predatory journals with very low impact factors. Whereas previously, most of scientific data was conveyed through a much smaller number of high impact factor journals with wide readership. Now we have this explosion of information, not only directed for scientists, but also for general populations. In the ones for general populations, they're written by journalists who are essentially popular news journalists. So they try to simplify scientific data, or they try to over-interpret data. And so this has expanded in the amount of confusion and white noise within the science as a profession. And all of this is happening and blowing up at a time when the world needs to hear and get leadership from scientific institutions like the WHO, like the CDC, like the NIH, but all of these have been undermined by certain governments. And on top of that, there's also been a big politicization of science, because previously, up until say the 90s, most of the high flyers in the scientific field came from the West, from the US, from Europe, Israel, Australia, South Africa, Japan, and Korea. Now with the rising scientific expertise in China, Russia, and other countries, a lot of these established Western scientific establishments are trying to maintain their dominance. And so they provide, or they are undermining or downgrading the accomplishments of these other countries. And so all of this, the result of all of this is to disunify what was previously a reasonably united front of scientists working together. And I think that all of this has contributed to the public's lack of respect and lack of trust of scientific experts. That's the basis of my talk. As for any solutions, I'm just an observer. I'm not a policymaker. I think that within the scientific establishment itself, there needs to be some kind of structure. But as long as scientific institutions are reliant on governments for funding, I think I don't know what they can do, what we can do. That's it. Thank you.
Thank you very much for your insight, Professor, even if it has ended on a sour note. And I want to say that this has been our last speaker for the day. However, that does not mean that our event is completely over, as in about 20 minutes we will be holding a bigger discussion panel or space, call it as you will, to which we invite all of you who participated, or even those who just watched but didn't have a chance to speak. Yet before we do, we'd also like to take this chance to thank you once again for joining us and participating in this event and sharing your knowledge, expertise, experience. We believe this international COVID-19 conference held online has been not only a place to share your ideas and to perhaps learn new things, but what we really aim is for it to hopefully become a real connecting platform for many various specialists from the fields of virology, immunology, as those are in fact rather wide fields with many different specializations and with a struggle such as the one we face against the COVID-19, both in terms of its scope, deadliness, but even the complexity or danger. It's very important that we can act to the best of our abilities and as quick as we were able, and combining all of that we know, what we've learned, what we've experienced across the world, that is the only way we see to resolve this problem as soon as possible. And that's why we hope that this conference won't be the end of such attempts, such struggles, that there will be many more, both held by us and maybe others, that the information within the scientific community will be really allowed to flow, opinions, experience, ideas, to pollinate with each other, to really just bring the solution to this problem. As part of that, we'll be hosting the discussion event in about 20 minutes from now at 11 p.m. Warsaw time, to which we once again invite all of you. Also, I'd like to remind that we, as the Foundation for the Support of International Projects, will be attempting to establish a working group of virology and immunology specialists to truly tackle this issue and do the work we all need to be done. Thank you very much.